Hi, I'm Maddie,
And I'm Michelle.
and we chose to study the eukaryotic elongation factor 1A2 gene.
We chose this gene due to its oncogenic properties. The gene was chosen due to its hypothesized involvement in ovarian cancer and breast cancer. Although it is also thought to be involved in pancreatic and lung cancer, the research we mainly looked at supports linkage to ovarian and breast cancer. This led researchers in study to study this gene because they saw potential linkage to cancer. It is thought that the overexpression of EEF1A2 is linked to estrogen receptor positivity due to its overexpression in breast tumors compared with normal breast tissue. This was also seen in ovarian tumors, but not in normal ovarian tissue. The gene EEF1A codes the protein elongation factor 1 alpha 2. The gene also codes for two proteins, EEF1A2 and EEF1A1, depicted in this image here. The two proteins are highly similar, 98%. They're two isoforms of this protein, and this actually makes it quite difficult for the researchers to isolate the proteins. However, they've been able to generate antibodies that allow them to distinguish between the two, and thus making EEF1A2 a quality candidate for oncology research. The eukaryotic translation elongation factor 1A is one of the key players in protein biosynthesis. Our gene encodes an isoform of the alpha subunit of the elongation factor 1 complex, which is responsible for the enzymatic delivery of amino acyl tRNAs to the ribosome. This isoform is expressed in brain, heart, and skeletal muscle, and the other isoform is expressed in brain, placenta, lung, liver, kidney, and pancreatic cells. These are normal healthy tissues and not tumor cells or tumor tissues. And our gene is also a member of the elongation factor 1 alpha gene family. Our gene was largely studied in humans, which can mostly be attributed to the fact that breast cancer is the most common cancer of females worldwide, and therefore the identification of changes in gene expression in breast tumors in comparison to normal breast tissue is greatly important for prognostic indicators and therapeutic targets. Studying the expression of our gene in human tumor cells was the first recognition of overexpression and the potential of our gene to have oncogenic properties. This is not limited to breast tissue, but also ovarian tissue and ovarian tumors as well. These studies were a great segue into further studying overexpression of our gene in mice. So mice are another organism. And this here, the NIH3T3, is the name of a fibroblastic cell that's found in mice, which one of the studies on ovarian tumors proved to be a good cell to use, a good cell line to use as an indicator for oncogenicity. Scientists are studying our gene in organisms such as humans and mice in hopes that it will shed new light on mechanisms of oncogenicity. The first evidence that our gene was found to play a role in tumor genesis was in 2002 in a study called Gene Encoding Protein Elongation Factor EEF1A2 is a putative oncogene in ovarian cancer. And this was a study that we focused on a lot in our research, and actually this image here came from that study, and this picture shows ovarian tumor cells showing the amplification of our gene. So the researchers were asking these questions of, is EEF1A2 and the process of protein elongation likely to be critical in the development of ovarian cancer? And why is EEF1A2 showing enhanced cell growth rate? Also, does overexpression of EEF1A2 induce tumor growth in vivo? Those are some of the questions that led to this study, and which we will dive into further next.
Historically, for a gene to be considered a tumor-promoting oncogene, it would have to be hyperactivated or overexpressed in primary human cancers and have the capacity to transform rodent cells cultured in vitro. Therefore, in the studies we researched, rat fibroblasts were used to explore why EEF1A2 was showing enhancing cell growth rates. In order to study this, the experimenters had to introduce the gene into the cell line. Frozen ovarian carcinoma tissue samples were taken and the gene was isolated using RNA purification. They grew fibroblasts from the mice in a cell culture, and the fibroblasts were transfected with 5 micrograms of the plasmid. These transfected cells were then incubated in solutions at 37 degrees Celsius for 14 days, and the agar plate medium was changed every 3 days to provide adequate nutrients for growth of the fibroblasts. The transfection efficiency was then determined by counting colonies that arose on the selective media. This showed which fibroblasts the plasmid had infected and could therefore be used to inject the genes into the mice. The rat fibroblasts are ideal for subcutaneously injecting EEF1A2 and inducing the tumor growth in vivo. To fully insert the gene into the mice, the experimenters injected the transfected fibroblasts into the hind legs of mice. This can be seen right here in these images. The parental did not have it injected, however, these 3 all had it injected, and you can see the tumor growth within. In the experiment, the gene's expression was controlled by the cytomegalovirus promoter, also known as the CMV. This is a constitutive mammalian promoter. Therefore, when the transfected gene was injected under the control of the CMV promoter, there will be constitutive protein expression within the organism. Experimenters would therefore be able to see the effects of the particular protein on the mice to determine its role in oncogenicity. The researchers were also able to compare the overexpression of our gene in the mice which displayed the tumor growth with the parental control mice that did not have any significant tumor growth observed. In any experiment, it is essential to understand how the experiment is working. In order for scientists to determine the expression of the gene EEF1A2, they originally observed increased expression in half of primary ovarian tumors. They induced overexpression by using a xenographic technique in mice by inserting a graft from a human into the mouse. In addition, the expression of the gene EEF1A2 made the fibroblasts tumorigenic and increased the growth rate of ovarian carcinoma cells in mice. The accelerated rate of growth factors showed overexpression was occurring. Western blots were used to prove indicative expression when comparing to parental control of tumors in mice. They did not inject the gene into mice and were able to compare the overexpression. Below you can see a gel electrophoresis, which was run to determine lines expressing EEF1A2. These lines show an increase in the EEF1A2 gene within the injected rats. The more dense areas seen right here in black indicate that the gene is present. This proves the overexpression of the gene, and the experimenters knew that their experiment was working and could be further analyzed to determine the functional role of the EEF1A2 gene.
As far as our gene's functional role, it is thought that it's involved in inhibiting apoptosis, involved in increased tumor growth rate, and involved in the process of protein elongation. You can see in the graphs here that E1, E2, E3, and E4 lines display more rapid and greater tumor growth than the parental and vector lines. Here's the parental, and here are the E1, E2, E3, E4, indicated by the dark shapes here. If you could remember back to a few slides earlier when I showed the image of the two isoforms of the protein, it's thought that the ability of both of those proteins to enhance cell growth and tumorigenesis could be related to their ability to inhibit apoptosis. And when apoptosis is inhibited, then thus tumor growth rate is accelerated. And we observed that clearly in the mice that grew tumors. You can see here in these images, as was shown before, these tumors are shown 20 days post-injection, growing large carcinogenic tumors. The EEF1A2 expressing NAH3T3 cells grow as tumors in nude mice, even though the parental cells do not. So how does a protein translation factor enhance tumorigenesis? Well, a protein initiation factor called E1F4E is highly expressed in primary breast carcinomas typically and established cell lines. It is thought to regulate tumorigenesis by enhancing the translation of genes promoting cellular growth. And EEF1A2 may function in the same manner, or is thought to function in this very same manner. Understanding the mechanism by which our gene, or EEF1A2, regulates cell transformation and tumorigenesis will be a very important part of our understanding of ovarian cancer.